Perfect. So I'd like to thank everyone for attending this webinar today. And so I'm going to talk a little bit about some work I did during my DPhil at Queen Mary in London, and then some work I've continued on here at Oxford. And that's mainly looking at Bruton's tyrosine kinase in macrophages and how it actually regulates innate inflammation in metabolic disease. So this audience probably doesn't need much introduction to diabetes, but it comes in two main flavors, type 1 and type 2. Type 1 characterized by insulin depletion due to a reduction of beta cell mass, and type 2 stemming from insulin resistance in the presence of elevated insulin levels, so reduced signaling through the IRS complexes. And globally, this is a huge problem, and I think it's been very much exemplified in the recent COVID-19 pandemic, where we know that patients who come in and present with diabetes are much higher and have a much more severe disease. And we know that around the world there's about nearly 300 million have diabetes of some description, and that number is set to double in the next 10 years. So this type 2 diabetes and obesity-driven diabetes is really becoming a global endemic, and there's an increasing prevalence in new economies and of populations of migrant populations in the Western world. And really, diabetes in itself is a manageable condition, but it's the microvascular complications that ensue later in life or as a result of diabetes that are really cause the problem. So nephropathy, neuropathy, and retinopathy. We know these morbidities cause severe mortality and motility in people. So I'm particularly interested in diabetic nephropathy. And diabetic disease occurs in about 20 to 30% of all patients with diabetes, and about 20% of all patients who require renal replacement therapy. And diabetes is a massive confounding in this. And annually it costs the NHS here in the UK about 1.5 billion. That's about 5 years ago. And in the States, 18 billion. And this number is set to increase because the supportive care that's needed for diabetic nephropathy, and especially renal replacement therapy, is high. So really, nephropathy comes as a multitude or as a result of lots of co-founders. So you've initially got your hyperglycemia, your hypercholesterolemia. And this causes a tissue insult, whether it's from metabolites, advanced glycated end products, increased oxidative stress, and ROS production. This all causes damage to the functional tissue within the kidney. Inflammation is also known to be a huge driver of tissue injury or damage within the kidneys specifically. And you get activations of many pathways, including NF-kappa B and the inflammasome. And this is a result of a huge infiltration of immune cells. Macrophages being one cell type that's actually very highly activated and correlated with disease severity. Inflammation is also correlated with the loss of and functional decline of cells, and you get an increase in fibrosis. And the real problem with diabetic nephropathy is that by the stage you get some discernible indication that you've got some disease pathology, you've lost about 70% of your functional kidney mass. So at this stage, you're left with a very small proportion of functioning kidney, and intervention is very difficult. So preemptive intervention is really key in this pathology. And really where my interest comes is in how to resolve this early inflammation that is maybe a driver of the loss of functional tissue within the kidney, and whether that's inflammation resolution or other factors that are pro-resolution in their effect. So drug repurposing and novel target validation. So one way that we as clinicians and as academics can look to find new medicines is through drug repositioning. So this is taking a medicine with a known target and using it for something else. And three things that we need to consider here is the target of the drug being tested is known. That is essential, but not always. The target of the drug is expressed in a cell type that's relevant to the disease. There's no point giving a drug if the target for that drug isn't actually relevant to the disease. And is there good tool compounds and medicines available to study this? So for drug repurposing, you don't necessarily need a medicine that's in the clinic. You can use medicines that have maybe made it to phase 2 or phase 3 in one indication and repurpose them for another indication where they may prove more beneficial. So I'm going to tell you a little bit today about Bruton's tyrosine kinase. So this is a tyrosine kinase. It's essential for B cell development. So you might wonder, well, B cells probably not the most, the cell type you think of straight away when you think of type 2 diabetes. But mutations in the BTK gene have been implicated in primary immunodeficiencies. And patients with XLA have normal B cells, but they don't produce mature B cells and have limited antibody function. And as a result, these patients are quite clinically vulnerable. So another indication where BTK inhibitors are used actually in the clinic is in chronic lymphatic leukemia and mantle cell lymphoma. So there's currently 2 that are EMA and FDA approved. They are ibrutinib, which is the first in class, and acalabrutinib, which is the second generation. And to my knowledge, there's 13 others that are in clinical trials for oncology, and some are currently in clinical trials for MS. So there's actually a wide variety of really good and validated target compounds here that could be used for repurposing. So BTK we know is expressed at high levels in B cells, but they're probably not the most relevant cell type in diabetes, or well, at least type 2 diabetes or obesity. But where else is BTK expressed? Well, here, this is using data from the BioGPS, which profiles all tissues and cells in the human cell types. And you can see here in CD19 positive B cells, you get high expression of BTK. But if you look down here in myeloid cells, you get relatively good expression. But in monocytes here, you get really high expression of BTK. And this is an interesting paradigm that we have a molecule that's well characterized in the adaptive immunity in B cells, but it's also highly expressed in monocytes, which are in the innate system and not so well characterized. And again, if we look in the mouse, in the same online repository, macrophages express high levels of BTK, along with osteoclasts, which are a subset of specialized macrophages. And again, microglia, and again, the spleen, where you expect to have high expression because there's a high B cell population. So this gives us a first bit of evidence that, BTK is expressed in a cell type, macrophages or monocytes, is actually clinically very relevant for inducing this pro-inflammatory milieu that we have in diabetes and type 2 and obesity. And there's some literature out there suggesting that BTK has known anti-inflammatory properties. So here's a paper that showed in cerebral ischemia that it regulates the NLRP3, so IL-1 production, and BTK regulates that. It also regulates LPS-stimulated macrophages, and we showed that in a model of multi-organ sepsis, so a really severe acute model of inflammation, that inhibition of BTK was beneficial. So these models are all very acute, so we wanted to have a look in a more chronic model, so the obesity diabetes model, whether you have activation of these pro-inflammatory pathways, which we know, and here's a molecule and a target that we can use to inhibit these pro-inflammatory pathways. So our central hypothesis in the work that in my lab is about limiting macrophage specific inflammation will be beneficial in metabolic inflammation and inflammatory diseases. And therefore, we sought to test the efficiency of BTK inhibitors of model of acute and chronic inflammation. And then we confirmed these in XID mice, which have a non-functioning BTK, but we're not going to show that data today. And this has allowed us to discover some really exciting biology around BTK in the innate system, which is kind of lacking in the literature at the moment. So first of all, we wanted to confirm that actually BTK is regulating macrophage inflammation. And we were able to do this assessing NF-kappa B activity and NLRP3 and inflammasome activity in both primary murine macrophages and human macrophages, data not shown. So here we have a really nice system. This is a reporter macrophage cell line, which secretes an alkaline phosphatase when NF-kappa B is activated. So we can activate NF-kappa B in our macrophages by stimulating them with LPS. We then pre-treated these macrophages with increasing doses of ibrutinib, which is a specific BTK inhibitor. And we could see that we had a dose-dependent inhibition of NF-kappa B activity in these macrophage cell line. So this was the first bit of evidence that actually BTK is specifically regulating NF-kappa B activity in macrophages. We then wanted to confirm this in a primary cell line. So we generated bone marrow derived macrophages. We cultured them in vitro for 7 days. And then we stimulated them again with LPS. And you can see with the red here, which is P65, it's cytosolically localized in non-stimulated BMDMs. But when you add LPS, it translocates from the cytosol into the nucleus, where it acts as a transcription factor for pro-inflammatory chemokines and cytokines in multiple inflammatory pathways. And in the presence of ibrutinib, you can see quite clearly that this inhibits the translocation of P65 to the nucleus, and the P65 remains cytosolically localized, indicating again in these primary macrophages that inhibition of BTK with ibrutinib was able to inhibit NF-kappa B activation. And this resulted, again, in our primary BMDMs, murine macrophages, when we increase the dose of ibrutinib in the presence of pro-inflammatory LPS, we had a dose-dependent inhibition of TNF secretion from these BMDMs, suggesting that if we inhibit BTK in macrophages, we are able to reduce the stimulation of TNF. And we know that TNF is one of those cytokines that's activated in chronic inflammation, including diabetes. And again, we were able to confirm this in BTK knockout mice, suggesting, again, showing that BTK knockout mice have reduced TNF secretion. We then went on to look at NF-kappa or NLRP3 inflammasome activity. And we were able to activate the inflammasome caspase-1 activity as a surrogate marker by stimulating macrophages with LPS and ATP. We were able to then stimulate them with increasing doses of ibrutinib, and in a dose-dependent manner, ibrutinib was able to inhibit NLRP3 inflammasome activity. And this resulted again in a dose-dependent inhibition of IL-1 secretion, and again, the same effect was seen in our BTK knockout mice. So again, telling us that BTK is able to regulate both NF-kappa B, pro-inflammatory inflammation in murine macrophages, and the NLRP3 inflammasome in primary murine macrophages. So this gives us an indication that here we've got a drug that we know that's in the clinic that we can use to target macrophage inflammation. So we wanted to move in vivo and look at whether BTK regulates macrophage inflammation in obesity induced diabetes. So for this, we use a very simple model of high-fat diet feeding mice for 10 weeks, for 12 weeks, sorry. And in the last 6 weeks, we treated them orally every day, 5 days a week with 2 increasing doses of ibrutinib. First of all, we looked at the 12 weeks period, we looked at insulin levels, and in our high-fat diet fed mice, we have an increase in insulin levels, which correlates with elevated blood glucose. And again, we're able to show that in our ibrutinib treated mice, we were able to reduce insulin levels and reduce glycemic load in these blood serum samples from high-fat diet fed mice. We then did a functional readout of glucose tolerance in these mice. So we fasted mice for 6 hours and then give them an oral glucose bolus of 2 grams per kilogram periorally. And then we did serial blood sampling after this. And you can see in our high-fat fed mice, you get an elevation in blood glucose, which stays elevated over the 2-hour time point of the window of this experiment. Whereas in our mice that were treated with the varying doses of ibrutinib chronically for 6 weeks, their blood glucose swiftly elevates and then returns to baseline within the 2-hour period. And this is quantified here with area under the curve. So we had significant improvements in glycemic control in our diabetic mice. So then we wanted to confirm that at the molecular level what was going on. And we knew that phosphorylation of IRS1 is a surrogate marker of insulin resistance in the liver. And again, in our high-fat diet-fed mice, we get increased phosphorylation of IRS1 in our diabetic mice, which is reduced in our mice that are fed a high-fat diet and treated chronically with ibrutinib. this also restored signaling through AKT and GSK3 beta in the livers of these mice. So this shows at a molecular level, we're able to improve insulin signaling and reduce insulin resistance in these diabetic mice chronically treated with a BTK inhibitor. So I'm particularly interested in inflammation in the kidney. So we wanted to see that what was going on in the kidney. We looked for BTK activation in the kidney, and indeed we get increased phosphorylation and activation of signaling BTK and its downstream effector phospholipase C. And this was abolished when we treated with ibrutinib. We then looked at mRNA levels in the kidneys. And again, we're able to see that you get an increase in Btk expression, which significantly correlated with CD68, a marker of myeloid cells, so infiltrating cells, and this was reduced in our ibrutinib treated mice. We then looked for some staining around the glomeruli for F480 positive macrophage, and again, in our high-fat fed mice, you get an increase of F480 staining, indicative of increased infiltration of macrophages, which was significantly reduced in our ibrutinib treated mice. And really the clincher here was that it actually improved renal function. So we knew reducing the infiltration of immune cells and inflammation in the liver directly correlates with improved renal dysfunction. So this is our albumin to creatinine ratio, which is significantly elevated in high-fat diet-fed mice. And again, at both doses of ibrutinib treatment, you were able to improve renal function. So here we have a drug that's available that you could give chronically, and it could improve glycemic control and renal function in diabetic mice. And again, we looked at our favorite inflammatory pathways, the NF-kappa B pathway. You get it in your high-fat diet-fed mice, you get an increase of translocation of the P65 subunit to the nucleus, which causes pro-inflammatory gene expression. And this is reduced in our ibrutinib treated mice. So here we're able to inhibit the activation of NF-kappa B in the diabetic kidney. When we looked at the NLRP3 inflammasome, again, you get an assembly of the inflammasome in our high-fat diet-fed mice. You get cleavage of gasdermin D, which is a pyroptosis marker, release of the active caspase-1, which causes cleavage and secretion of IL-1 beta. And this is all reduced in our ibrutinib treated mice. So here we show that in the tissue in these diabetic mice that we've inhibited the activation of the NLRP3 inflammasome. So just to sum up my talk today, and I think I'm probably a bit under time, but that's good. That BTK does regulate macrophage inflammation in obesity-induced diabetes. It restores signaling through the IRS1 complex, improving glycemic control. It protects high-fat diet-fed mice from the development of diabetic nephropathy, and it does this by reducing inflammation, specifically NF-kappa B and the NLRP3 inflammasome in the diabetic kidney. So overall, I think I've shown that inhibition of Btk improves glycemic control. It reduces immune cell infiltration into the diabetic kidney. It reduces activation of these two pro-inflammatory pathways, NF-kappa B and the NLRP3 inflammasome, resulting in improved renal function. And we propose that this is specifically through inhibition of BTK expressing macrophages. And with that, I'd just like to thank all the other people involved in this, from the British Heart Foundation, who funded my DPhil at the William Harvey at Queen Mary in London, and who continue to fund me here in Oxford. David Greaves, my supervisor, Keith Channon, our collaborators in Italy who do all our Western blots, they're fantastic. And for the other people who provided money for this project. And with that, I'd like to open the floor for any questions. And thank you for listening.
Thank you so much for your valuable presentation, Doctor. And now the session is open for questions. If anyone had any questions, please drop your questions in the chat box, or else unmute your audio and shoot your questions to Dr. Gareth Purvis. I repeat, if anyone had any questions, please drop your questions in the chat box. Besides, we will also leave the mail ID of Dr. Gareth Purvis so that you can drop a mail to Dr. Gareth Purvis in direct. Okay, it's like no one has any questions. Besides
Professor
Dr.
Edwards,
Gareth,
could you unmute your
you had a
microphone?
question?
Can you unmute your
Sorry?
microphone, Professor Edwards?
Okay, okay. Can you hear me?
Perfect. Yes.
Okay. Well, first of all, Gareth, a very good presentation, much enjoyed, really very interesting. Could you tell us a little bit about the question of whether there might be side effects? Because obviously, as you rightly say, it's been used, in fact, in chronic lymphatic leukemia, etc. But clear, if you're going to be using this, in fact, in the early treatment of diabetes and in the metabolic syndrome, have we got any kind of idea what might be the more perhaps problematic side effects?
So ibrutinib in itself is quite a dirty kinase inhibitor. It targets quite a lot, and it's known to cause atrial fibrillation. But it's been shown in a recent blood paper that's due to C-terminal kinase activation, and the more modern BTK inhibitors don't have this side effect. Another one would be that you're sort of dampening your immune response overall, so you could be more prone to infection.
Exactly.
So this is kind of it's a more proof of concept. I think that the chronicity factor of using these inhibitors is probably not viable in the clinic, but for really short-term anti-inflammatory, I think we might be onto something, and that's where my research is more heading in that direction. We've shown in polymicrobial sepsis in mice that it improves the survival. So we think maybe in short doses, like you give a steroid, this might be another anti-inflammatory that we have in the arsenal that maybe is untapped.
Can I just follow that up with a second question? And that is that I know the work of Professor Schwartz, in fact, in Yale, if you know that, but he showed very beautifully that the activation of the inflammatory pathway in blood vessels was very dependent, in fact, actually on shear stress, and that if you had normal blood vessels, then in fact you wouldn't get NF-kappa B activation. But if you replace the basement membrane of collagen, in fact, with fibronectin, which is what happens in diabetes and happens in cardiovascular disease, then you get this very marked NF-kappa B response. And so I just wonder whether your work might be really quite helpful in that particular area.
I think it's something that's definitely worth investigating for sure. And shear stress is one of the precursors for, athero load and everything. So, those areas of turbulent stress. So yeah, I think if you could reduce the activation of those adhesion molecules, again, that would be beneficial. And if that's NF-kappa B dependent, more the merrier for using a BTK inhibitor. We've actually done the athero models and shown that we reduce burden and macrophage load within the plaque. So that's something we're working on in collaboration with Keith Channon and Claudia Monaco here in Oxford. So, but again, we're thinking of moving more to the acute inflammatory. Cool. Thank you very much.